will be presenting Implementing an Effective Listeria Control Plan for Dairy Products. Dr. Marshall is the Chief Scientific Officer for Microbiology at Eurofins. He has over 20 years experience in academia and industry. He currently holds adjunct professor positions with Colorado State University and Florida State College. With over 250 publications and over 125 invited presentations, his scientific research and outreach interests focus on improving the microbiological quality and safety of food. We're excited that he's here with us today and sharing his extensive expertise on this topic. But before we get started, we want to go ahead and go over a few housekeeping items. This webinar is being recorded, and we'll be sending out the recording via email along with the slides from the presentation today within the next couple business days. All the questions that come in throughout the presentation will be answered after Doug completes his presentation. And any questions that we aren't able to get to during the time, we will follow up with following the webinar. To submit a question, you can go over to the questions panel in the dashboard over here. And if you just type your question in and hit send, we'll get that and address them at the end of the presentation. So with that, I'll go ahead and turn things over to Doug, and we can get started.
Okay, thank you, Heather. I appreciate everyone's time today in spending a few minutes with us talking about Listeria control in dairy plants. And what I want to first of all tell you is that I started working with Listeria back when it was first discovered in the early 80s. And at the conclusion of this talk, I'll be sharing a few slides of some things we discovered back then that are still relevant today. So most of you know who Eurofins is. I just want to briefly tell you that Eurofins' corporate mission is to improve the health and quality of life for consumers as well as our customers. In most of the market segments that we participate in, we are #1 in the world. That includes both food and environmental testing. In terms of locations, we have a number of locations in the U.S. That deal with foods. The one in yellow are grain locations. The one in red would be food chemistry or food microbiology laboratory testing facilities. My view on how we work with food safety is captured in this paradigm. Obviously, since I work for a food testing laboratory business, at the top of the list is rapid and specific detection of food issues, primarily pathogens in today's study case. And so you use detection and testing in order to inform pre-harvest controls. So in other words, what happens on the dairy farm can have an impact on processors' ability to deal with Listeria. We also use this test data to inform us in terms of post-harvesting controls during processing. And then both of those applications then, we also do a subsequent follow-up test to measure the efficacy of those controls. And that feeds into a risk assessment that then tells us what kind of behavior changes we need to deploy in our facilities. Brief background on what Listeria monocytogenes is. It's a gram-positive, non-spore-forming, rod-shaped bacterium. It is facultatively anaerobic, meaning it can grow with or without the presence of oxygen. So in many dairy products, finished dairy products, vacuum packaging is used to control mold growth and spoilage, but it does not have much of an impact on slowing the growth of Listeria monocytogenes. Unlike most of the other mesophilic pathogens that you're aware of, such as Salmonella and E. coli, Listeria monocytogenes is a psychotrophic bacterium, meaning it prefers to grow in mesophilic temperatures, in other words, around warm body temperatures, but it can also multiply at refrigeration temperatures. Fortunately, the growth rate is slower at chill temperatures, but chill storage in and of itself is not a very effective control measure. It is generally sensitive to heat processing, so the pasteurization measures that are deployed in the dairy industry will kill the organism. It is found in healthy cows, raw milk, and in dairy processing environments. Because of its widespread nature, then, it behooves the industry to have a Listeria control program to make sure that finished products are not contaminated. It's also known to survive cheese ripening processes, not all, but some. And many of the foodborne outbreaks that have been associated with Listeria monocytogenes have been associated with consumption of contaminated cheese. The first known foodborne outbreak was due to contaminated sheep manure being used to fertilize a cabbage patch, that then the cabbage was processed into coleslaw, and because coleslaw had a long refrigerated shelf life, the bacterium was able to multiply and grow and reach infectious doses. According to CDC, listeriosis is a disease that is caused by Listeria monocytogenes. It is generally very rare. Only about 1,600 individuals will get listeriosis a year. The problem, however, is that it has a very high mortality rate, generally considered to be one of the highest mortality rates of all foodborne bacterial infections. Those infections are most predominant in high-risk individuals, such as pregnant women and their fetuses, as well as the newborn, and also immunocompromised individuals, in other words, those that are taking immunosuppressive drugs or have some underlying condition that renders their immune system not as vigorous as it should be. And because of these conditions, then, public health authorities have issued an advisement that pregnant women are to avoid certain kinds of cheeses and unpasteurized dairy products, and some of these are listed here. So that's not really a good PR campaign for individuals manufacturing these kinds of products. Today's talk is focused on dairy products, but remember that Listeria monocytogenes also has the potential to be found in lots of other food products. So if you're co-manufacturing other food products, please consider that the bacteria may be an issue there as well. So we've had deadly foodborne outbreaks of Listeria associated with fresh produce, processed meat products, as well as dairy products. Some recent listeriosis outbreaks associated with dairy foods. I managed to carve these out of some CDC data. So just in the last couple of years, there have been 4 outbreaks associated with dairy products. Those are listed there. And before 2012, there were multiple outbreaks due to raw and unpasteurized soft-style cheeses. And there was one small outbreak attributed to consumption of contaminated pasteurized milk. Put this slide in to try and give you a feeling for what Listeria monocytogenes has to do to be able to survive in the environment, in a product, and in the gastrointestinal tract of an infected human. There are lots and lots of challenging environmental conditions that the organism has to address. On the left side of the figure, these are those that are in the product, and I'll address a few of these as we go along. In terms of challenges in the GI tract, those are just there for your own edification. So what then are some contributing factors then to Listeria monocytogenes causing outbreaks? And in addition, remember that Listeria presence in environmental samples, particularly food contact surfaces, or in finished product is a leading cause of recalls as well in the sector. So inadequate thermal processing is one contributing factor. A refrigeration temperature that is too high, or a refrigeration product shelf life that is too long. Inadequate product flow through the process, in other words, where there is an opportunity for cross-contact with raw materials. Inadequate personal hygiene, where individuals who may have direct product contact or contact with direct product contact surfaces might be inoculating Listeria monocytogenes on a product. Inadequate cleaning and sanitation, and subsequently inadequate environmental monitoring to be able to validate that your cleaning and sanitation program is working, and then to be able to verify that it's working every time that it's implemented. And then inadequate end product testing. So these are all contributing factors, and we can drill into these in a little more detail. In terms of inadequate thermal processing, as you know, is a time temperature process. So the issues that we've seen are going to be a process temperature that is too low or a process time that is too short. What we're not dealing with here is the possibility for cross-contamination after thermal processing. Refrigeration temperature too low. There have been many studies that have been published over the years that have demonstrated that refrigeration units, whether it's during transportation, at retail, or in consumer households, those temperatures generally are not held at a permissive temperature. In other words, no more than 10 degrees C. So some data that I found is that a majority of the household units have temperatures exceeding 9 degrees, and about a third of the retail store units also have temperatures that are violate the food code's chill holding temperature requirements. To demonstrate to you how that increase in storage temperature impacts the multiplication of Listeria, these are some data we generated quite a while ago that shows that once you get past that temperature threshold of 10 degrees C, in this case 11 degrees, the bacterium reached its maximum population level within 6 days. So many of the refrigerated shelf life of dairy products far exceed 6 days. So you could have Listeria reaching maximum population level in those before the end of shelf life. Other contributing factors include inadequate cleaning and sanitation. So obviously this is going to be a battle between production and QC. I've had many discussions with individuals about the use of floor drains as a spot indicator of whether or not you've got Listeria in your plant. And in most dairy processing plants, you will find Listeria or Listeria monocytogenes at the floor drains. What maybe matters more than finding Listeria there is there an opportunity to get Listeria out of those hot spots and more widely disseminated in the processing plant. And so again, you need to think about, are there point sources of inoculation of Listeria that you're not getting effective control during cleaning and sanitation? To turn the coin over on that, are there point sources of inoculation that during cleaning and sanitation you are disrupting those locations and then widely disseminating Listeria in the processing plant during those cleaning and sanitation operations? And a couple of examples that I've seen here are the use of high-pressure water sprays during cleaning. Again, as soon as those sprays hit a hot spot, you can aerosolize the bacterium and send it throughout the manufacturing facility. Another opportunity here is using high-pressure air hoses. Again, those work great to move debris off of equipment, but they're also very good at disseminating microorganisms throughout a facility. Inadequate product flow is another potential example. And in this case, it generally breaks down into two root causes: failure to segregate pasteurized products from raw materials, and then also a failure to segregate employees who work in the finished product area and preventing them from working in the raw product area. So one way you can deal with this is to map out both the product flow as well as employee flow, and also remember that there is equipment that travels along with those employees. So make sure you're mapping the travel of that equipment as well. And what you really need to be looking for are areas in which cross-contamination can occur. Some direct food product contact surfaces that have been known to harbor Listeria monocytogenes are listed here. Obviously, if you're a fluid milk producer, your pasteurization step is going to be the definitive kill step. And you're obligated then to make sure you don't cross-contaminate the product between the pasteurizer and the final package closure steps. So any place that that product is exposed into the environment, such as at the filler heads, is an opportunity then for Listeria to get back into the product. This also includes packaging systems for finished products. Any conveyors that may be moving finished products such as cheeses, as well as unclosed containers. Chilling solutions also have been known to harbor Listeria, so you need to make sure you've got those under control either through routine monitoring or through the use of sanitizers in those solutions. Slicers, dicers, shredders, and whatnot, these particle size reducing unit operations oftentimes are very complicated pieces of equipment. If they are not broken down during the cleaning and sanitation process, there are many opportunities and many harbored sites in those pieces of equipment for Listeria to gain attachment on those surfaces and perhaps even proliferate and grow at high population levels. Collators and assemblers, product movement items, spiral coolers and blast freezers have one unique feature that makes them rather challenging. Number one, again, they're large, complex pieces of equipment that are difficult to clean and sanitize. Number two, they are using air as a component of the cooling process. And if that air is contaminated, again, you could be blowing Listeria monocytogenes right on the finished product. And then hand tools, gloves, and aprons also are known to be focal points for Listeria cross-contamination. Additional contributing factors include inadequate personal hygiene. This includes both clothing and outerwear, as well as hand washing. Maintenance personnel frequently are omitted from personal hygiene requirements, and that's unfortunate because they can be an easy vector to transport Listeria from a highly contaminated area to a highly uncontaminated area, and making those equipment maintenance personnel good vehicles for transporting the organism. Hand washing, there are many who recommend using an alcohol-based wipe for hand washing. And then also the use of iodophor hand dips can be an effective control measure. Okay, shelf life too long. I've already demonstrated to you that refrigeration, although it does slow the rate of growth of the organism, if refrigeration is inadequate, Listeria monocytogenes can multiply and grow and reach high population levels before the end of shelf life. So what the regulatory agencies are starting to ask more questions about is, what is your product shelf life determined by? Is it determined by spoilage, or is it determined by safety? And so if you are manufacturing products that have the potential for contamination with Listeria monocytogenes, perhaps your risk assessment should include whether or not your product shelf life is adequate to prevent high population levels of Listeria from occurring should contamination happen. And one way to do that is to run an L monocytogenes challenge test on each product to be able to answer that question. FDA, in their preventative controls proposed rule, has a section on environmental monitoring, and their rationale is many of the Listeria monocytogenes outbreaks have been due to poor environmental control of the bacterium, and it wasn't a breakdown of the HACCP plan, so the kill steps were working. It's just in those facilities there was the opportunity for cross-contaminating finished product with the organism, and usually a long refrigerated shelf life that allowed the bacterium to multiply and grow. So in this case, an adequate environmental monitoring is a key control component for Listeria monocytogenes in dairy processing facilities. When I teach environmental monitoring, my philosophy is this should be a search and destroy mission. You need to invest your testing dollars to find hotspots in your facility, find ways to control those hot spots and make sure that the effectiveness of your control programs are working every day, every week. And the only way you can do that is to search and destroy for those hot spots. Many people advocate the use of Listeria species as your test target, particularly on direct food product contact surfaces, because if you find Listeria, there's not necessarily a recall in order. If you're testing direct product contact surfaces for Listeria monocytogenes, and you find Listeria monocytogenes on a direct product contact surface, then the assumption is the product also is contaminated, and now you have a recall event. Some common environmental harbingers for Listeria monocytogenes are listed here. These include equipment framework. And again, if you put your mindset in, if I'm Listeria and I want to find a place to hide in your manufacturing facility, where would that be? Would it be on an easily clean plate of stainless steel or on an easy to clean wall or floor? Or would it be in a nook or cranny or an area where food, moisture, and warm temperatures are prevalent, because that's what Listeria needs to multiply and grow. So you're looking for these nooks and crannies to find Listeria. Those are also areas that are difficult to obtain a sample. So you have to be creative. I've used little narrow spatulas to be able to pull off biofilms from some of these nooks and crannies. I've used a telescopic gun barrel cleaner to be able to scrape off debris from areas and equipment that are difficult to gain access to. Okay, floors and drain, we've already talked about this. Any place where there's standing water, that would include underneath foot baths and around foot baths, by the way. Walls, ceilings, other overhead equipment. Any place where there's condensate that has the ability to form as well as to fall onto product contact surfaces. A good place to look is exposed wet insulation around piping, also in walls. Forklifts, trolleys, other kinds of transport vehicles that are used to move product in and out of the facility. I've even found Listeria monocytogenes on cleaning materials. Mop heads are a classic place to find Listeria, and also on maintenance tools. Additional areas are conveyors. The belts and rollers of these complicated pieces of equipment are very hard to clean if they're not thoroughly broken down and doing a soap cleaning and sanitation process. Control panels and switches, rubber door seals and equipment, particularly those that the gaskets are cracked or worn. Trash cans, inline air filters, motor and pump housings, cracked hoses, and ice makers. Okay, in-product testing is an important part of a Listeria monocytogenes control program. I have had many lively debates with manufacturers about the need for in-product testing, and their side of the argument goes like this. If my hazard program is working, I know that my kill step is effective, and I know I have a history of doing in-product testing with no positive detections, why should I be wasting my money on doing in-product testing? So that's a very logical, very sound argument. And obviously I work for a testing company, so there is some bias in this. But imagine if your product is involved in either a recall or an outbreak, what is your rebuttal when you were dealing with your lawyers? Well, we didn't do any in-product testing because we didn't feel like we had to. So that's not a very effective and not a very convincing rebuttal when you're challenged with those kinds of questions. I will agree that in-product testing of finished products is usually useful to detect gross contamination, and it is not very useful to detect spot contamination events. So there are some deficiencies in terms of the usefulness of end product testing. It's up to you to decide whether the risk of not testing is worth it or not. Most people do have to do end product testing because that's a requirement to sell their product. Product testing should also be done to assess the risk of rework for Listeria monocytogenes rather than Listeria. Because if you find Listeria, the assumption is it has the potential to be monocytogenes and you would have to recall it anyway. So according to FSMA, these are the required food safety plan elements. You can just go around starting at performing a hazard analysis, and then you go through the cycle of programmatic elements, and all of these need to be supported by records. And I think it is FDA's intention that those records would include test results. And that's how you can best equip yourself when questioned whether or not your programs are working, is when you have test results. Those are the ultimate pieces of scientific information concluding that your products and your process is safe. So when you're doing a Listeria control program reassessment, these are some elements that I think are valuable to include. First off are the inherent levels of Listeria monocytogenes in your raw materials and ingredients. The efficacy of your kill steps. So in this case, many companies now are going back and reassessing their kill steps, doing challenge studies to be able to prove that their kill steps, in fact, do eliminate Listeria monocytogenes. Looking at your post-kill controls, particularly products that are exposed to the environment between the kill step and the final package closure step. Looking at your shelf life and asking what is the potential of Listeria monocytogenes to either survive and or grow during the intended product shelf life. Making sure you are routinely looking at your environmental monitoring records, doing trend tracking, root cause analysis if you have contamination events, and then having an effective reassessment of whether or not your controls are working, and then also your finished product testing records. So in terms of your kill steps, there are several ways in which you can validate whether or not those indeed are effective against Listeria. You can do definitive challenge testing. You can use computer monitoring. You can find a history of scientific literature, or you can consult experts who can give you an expert opinion. In terms of your post-kill control steps, number one, I would ask, do you know your source of contamination? And secondly, if you know where those are, do you have effective control measures to keep those locations free from contamination? So here's an example of a secondary control measure in many dairy products that has the ability to impact the growth of Listeria during refrigerated storage. So in this case, it's just simply the amount of lactic acid present in a specimen. And what you can see is the amount of lactic acid is highly determinant in terms of the ability of Listeria to grow. So concentrations of 1% Listeria monocytogenes can grow at a bit slower rate than without lactic acid. But once you get above 1.5%, the bacterium will not grow. So that is an effective control step that many of you have in your products. Okay, I want to turn now and spend the last part of the webinar talking about other microorganisms in the environment that have an ability to impact the ability of Listeria monocytogenes to survive in that environment. And it'll be a little story I want to share with you. The organism I want to talk about is Pseudomonas. Many of you know this in dairy products because it is a predominant spoilage organism in many kinds of dairy products. Listeria is a bit different than Pseudomonas because Pseudomonas is a gram-negative non-spore forming rod. It has a number of metapodically diverse species. Most of these are aerobic, meaning they absolutely need oxygen for growth. So vacuum packaging is a good way to control their multiplication in finished products. Like Listeria, Pseudomonas is psychotrophic, meaning it grows well at refrigeration temperatures. And it produces, because it's a predominant spoilage organism, the reason why it's a spoilage organism is because of its metabolism. There are many proteolytic and lipolytic strains that can cause proteolytic spoilage. This could lead to the formation of putrid aromas, hydrogen sulfide like aromas. And many also are lipolytic, which can lead to rancidity-like spoilage defects. Many of the pseudomonads also are prolific exopolysaccharide excretors. And because of that, they are one of the primary biofilm-forming bacteria in food processing environments. A few of the pseudomonads are opportunistic wound and respiratory pathogens, but they're not considered to be foodborne pathogens because they generally do not cause gastrointestinal distress. So I mentioned that Pseudomonas is a primary biofilm former, and this is the general cycle at which biofilms are formed. So you take dispersed, free-floating individual cells. If they land on a surface, and here we're going to go left to right on your screen, what happens is as soon as those cells land on a surface, this could be a pristine 304 piece of stainless steel. Those cells are going to immediately attach to those surfaces, and they attach via van der Waals forces in hydrophobic and hydrostatic interactions. And even after just a very quick attachment time, it's very difficult to remove those cells by physical force, meaning by shaking or by running water across that surface. Once those cells become attached, and if there is an abundance of moisture, water, and a permissive temperature, then those cells can multiply, and you get what are called microcolony formation. That's the second blob. As that material ages and matures, you get a large multi-cell biofilm that could have channels running through that channel nutrients and water into the biofilm and channel waste materials out of the biofilm. So it actually acts much like a very primitive organism, multicellular organism. And then as the biofilm further ages, then when you are applying your remediation steps through cleaning and sanitation, you may be doing a good job killing the cells from the outside of that biofilm. But unless you have complete physical removal of that biofilm, you're going to have a residual population deeply embedded in that biofilm that could continue to multiply and grow and redevelop that complex biofilm. And that makes controlling these very, very difficult. For those of you who haven't actually physically seen a biofilm, I would ask you to go into your bathroom and look in your tub and tell me what you see in that tub. If you see any black material or gray material, what you're looking at there is actually visual biofilms. And you certainly don't want those in your manufacturing facilities. So this is what happens when you take Listeria and put it on a piece of stainless steel. This was put on stainless steel, and then after 30-second attachment time, and that piece of stainless steel was put under running water for 30 seconds. And what you can see, these are the remaining cells that are still firmly attached to that surface. They were not easily washed off. As those cells then begin to proliferate and grow, you get what's called microcolony developments, and then you get further development, and you can get a complete multi-layered mass of bacteria. So this is what a biofilm looks like under the microscope. And when you get closer to a well-aged, well-developed biofilm, you can see all this multicellular exopolysaccharide and proteinaceous material that is attached and kind of glues these cells together. This is actually what Pseudomonas looks like. This is not Listeria. But if Listeria gets entrapped in that kind of matrix, you can see how difficult it is to control that bacterium. Now, I want to talk about the relationship between Pseudomonas and Listeria. Biological interactions, also called synergism, come in a number of different flavors. So I'm going to use these cartoons to kind of illustrate what that is. So neutralism is a situation where if the two organisms are present in a habitat, they don't care about each other. Neither one is affected by the other. Mentalism is a situation where one of the organisms is benefited and the other organism is severely detrimented. Competition is a situation where the organisms are both competing for the environmental niche, and neither is benefited by the association. Antagonism is where one organism is benefited at the expense of another. Mutualism is a situation where both organisms are benefited by the relationship. Hopefully your interaction with your home life fits into the mutualism category. And then commensalism is a situation where one organism is benefited by the interaction where the other one doesn't care. In the case of Listeria and Pseudomonas, this is the interaction that they have in a dairy processing plant. What does that look like? Well, in this case, over 20 years ago, we looked at this relationship between Listeria and Pseudomonas. At the time, Listeria was first discovered, and the scientific community was saying that Listeria was a poor competitor in the environment. Well, that didn't pass the sniff test to me, because if it was a poor competitor in the environment, why are we finding it all over the place in the dairy processing plant? And one of the things that we looked at was not solely looking at Listeria alone, but how does it interact with another very common spoilage organism, and that's Pseudomonas. And what we found was that when Pseudomonas was present in milk and had an opportunity to grow a little bit, when you put Listeria in there, its growth rate was stimulated by the presence of Pseudomonas. We didn't know why, but we just knew that Listeria was very happy in the presence of Pseudomonas. What we also found was that Pseudomonas was not affected by the presence of Listeria, so it went on with life as if Listeria wasn't there. That's the commensal relationship. To show you how impactful this relationship is when we inoculated whole milk, skim milk, or non-fat, reconstituted dry fat milk. When we added Pseudomonas, grew it up overnight, and then added Listeria, Listeria grew much better, in this case a shorter generation time, when in the presence of Pseudomonas than without. We then wanted to ask the question, why was Listeria growing better in the presence of Pseudomonas? And so we looked at some metabolic factors of Pseudomonas that might impact it. So we measured whether or not the two organisms were able to carry out lipolytic metabolism, and indeed both organisms were able to hydrolyze lipids. We then asked, is proteolysis a potential factor? And in this case, Listeria monocytogenes is a non-proteolytic organism. So for it to actively grow in an environment, it needs peptides and amino acids in that growth environment. And what we were able to demonstrate is that Pseudomonas, in this case, was able to produce those proteolytic byproducts. When we artificially hydrolyzed milk protein and then added Listeria to mimic what Pseudomonas does, we indeed found that that generation time was significantly faster when Listeria was grown in milk where protein hydrolysis had occurred. So what does that mean then for you in the dairy processing industry? Well, what it means is anything you can do to affect control the biofilms in your plant that's controlling both Pseudomonas and Listeria, I think you'll have a much better and more effective plan in keeping Listeria under control when you do not have these proliferative biofilms present. So Heather, that concludes the formality of my talk. I'll let you take over the session, and we have a few minutes for some Q&A.
Great, thanks, Doug. Give me just a second here. Okay, so we have a few questions that came in throughout the presentation. First, I wanted to start with, Doug, what would you say is the biggest avenue for Listeria entering a plant?
Okay, I think there are a couple of things that come to mind here. Number one is raw materials. So anytime you're bringing in contaminated raw materials, such as raw milk, for example, will have Listeria monocytogenes, and the tanker trucks at which those are being transported. Those are going to be a vehicle both to bring the milk in as well as bring Listeria onto the operating plant premises. The second area would be employees being able to track Listeria from areas outside of your control. This would be primarily outside the processing plant. So any individuals that are coming in your parking lots, walking into the plant, when they're going out on break, if you do not have a captive garment program that includes shoes and proper sanitation, then those employees have the potential to track Listeria monocytogenes into the plant. Other areas, if you go back to just general GMPs, if you have improper vermin control, those vermin can be a source of Listeria monocytogenes either in their intestinal tracts or on the outsides of their body. And then anytime you have equipment running back and forth in the plant, when you look at how that equipment traffic is moving, if it is going from a raw area to a finished product area, it's a wonderful opportunity to be able to bring Listeria into those sensitive control areas. Next, please.
Okay. All right. Next question. Is granular clot ammonium effective versus the liquid type? And this would be for drains, pits, and cooler pits.
Okay. The question is related to the type of chemical sanitizer being used. The granular sanitizers, to my knowledge, the only way they're going to really be active against Listeria is when you have the ability to hydrate those granules to be able to get access to the cells. So I know that the granule areas are being used just as broadcast media on high traffic floors. But in terms of the specific application in the floor drains, you really need to, number one, work very closely with your chemical supply house, and number two, actually do the microbiology testing to be able to get definitive data by which to answer that question in your own facility. Next, please.
Okay. Next question is, how can you determine the quantity of swabs needed in your environmental monitoring program, and can you base it on your square footage?
Good question. When I teach environmental monitoring, if also you are interested in having a guide, please let us know. I'd be happy to send that out. Again, there's a couple of ways you can do this. I prefer to do environmental monitoring on a risk basis. So invest your testing dollars early in the program to be able to identify those hotspots, and then make sure you are doing very routine testing of those hotspots on a regular basis to ensure control. Some people advocate using a square footage, and what that does is it makes your selection process easier, but it also may force you to reduce the amount of testing you're doing on your hotspots. So there is an old formula that's used that I have in some of my EMP guides that was one that was used by the pharmaceutical industry in order to certify a processing room to be able to bring back into production. And what you do is you just take the square footage of the area in your facility in meters squared and take the square root of that, and then that will give you a number that then you can use as the number of swabs to do whenever you do your environmental monitoring. So you're more than welcome to do that. Just remember that that's not a risk-based environmental monitoring program. That just simply is a mathematical base, and you may be under-testing your hot spots and over-testing your clean spots. Next, please.
Great. I think this is the last question that I have, unless anybody has any additional, please get those in before we close. So last question I have here is, can you recommend any good reference books or materials about vectoring?
Yes. When everyone gets the slide deck, there is a reference to an article in a Dairy, Food, and Environmental Sanitation journal that was, the primary author was Tompkin. So I've listed that reference on a couple of slides. That would be a good reference paper for you, because it really highlights a lot of the areas that are primary Listeria issues in dairy processing plants.
All right. Well, great. That's all the questions that I have that have come in. If you guys have any additional questions that you think of after the webinar, please feel free to reach out to us via the contact information on the screen or by replying to the email that we send with the recording and the slides. So just as I said, as a reminder, we're going to send the recording and the slides out within the next business day or two. You should receive it by the end of the week. And if you have any more questions, please let us know. Otherwise, thank you very much for joining us today, and thank you, Doug, for presenting this information. And we'll see you next time.
My pleasure, Heather. Thank you, everyone, for your time. Have a nice day.
Thank you.